100% healed.
What have people healed by COVID done? What information have they healed used? Advanced science explains it to us. Let's understand it with immunopathology and epigenetic naturology. In 2019, an RNA virus called coronavirus appeared. Coronavirus is named after growths called spikes, which allow it to hook into our cells. To understand how 100% of patients are healed, we need to answer these questions. The keys to healing. What prevents the virus from entering the body? What hinders the entry of the virus into the cells? What were the formation of cellular agglomerates? What hinders coagulation and what protects the vessels and capillaries? What blocks the viral influence on the nerves? What improves expectoration and stimulates the defenses? What counteracts inflammation? What to recover the taste? Nicotinics. How to block the reactions of the body against itself. 0. How to combat visceral obesity and the excess of sugars that favor viral disease. To understand how 100% of the sick are healed, we ask the above questions in understandable scientific language. For a doctor or biologist, the keys to healing. What hinders the entry of the corner? What hinders the cell locks against the virus? What hinders the formation of cellular syncytia? What hinders coagulation and protects vessels and capillaries? What to compete with the nicotinic receptors, apolifunctional symbol alpha 7 and apolifunctional symbol alpha 9? What improves expectoration and stimulates the defenses? What counteracts inflammation? What the taste recovers? Nicotinic receptors. How to block molecular mimicry. 0. Metabolism. How to defeat the metabolic syndrome, the cytokine storm and free radicals. Premises. We understand what happens. This is to answer points 1 and 2. What is a virus? What the virus wants. How the virus penetrates. What is the spike? What are ACE2 and CD26 receptors? Is the virus a living being?
No.
No. The virus is comparable to a USB stick. It contains data and programs that cannot function without an external cell. The virus is harmless. Disease is the body's reaction to its membrane proteins, which produces noxious reactions. What does the virus want? The virus wants to reproduce and survive. That is to maintain and protect your data over time, multiplying everywhere in as many copies as possible. If possible, he tries to insert his data into the genetic code of the human cells or the bacteria that populate it to use them for his projects unknown to us. What is the spike? The spike is a glycoprotein. Spikes are like keys to the virus that enter special cell locks. These keys allow the virus to penetrate inside the cells and use the biochemical abilities of the same cells to reproduce. The spike, the virus to enter the cell must pass through doors presenting a path, a key, the spike, in a cellular lock. The cell locks are called ACE2 and CD26, or DPP4. The body must learn to live with the virus by modulating its immune response. Vaccination would have this goal. The body must calibrate its specific defense reactions to avoid self-harm. If the body's defense is excessive or inadequate, exaggerated reactions are produced that can trigger what is called the cytokine storm, which, if not controlled, can even lead to death. Let's try to better understand how the virus enters cells and how it can cause damage. The cell locks, ACE2, are the most involved locks. ACE2 are mostly present in the lungs, nose and intestines. In the lungs they are present in type 2 pneumocytes, i.e. the cells that line and keep the alveoli dilated and allow the exchange of respiratory gases. So if the excessive antibody reaction is triggered, you can no longer breathe well. In the nasal passages they are present in the goblet cells, i.e. those that secrete mucus, so here are the signs of a cold. In the intestine, they are present in enterocytes, i.e. the cells responsible for the absorption of nutrients, which is why we lose weight. This affinity, as we have seen, is due to the bond between its spike protein and the receptor on the cell membrane called ACE2. ACE2 uses a protein produced by cells which helps the virus to enter. It is a cellular enzyme called TMPRSS2 that helps activate the spike protein. CD26 glycoprotein, or DPP4, General cellular lock. The first receptor of the virus is the CD26 glycoprotein. It is present on all cells. It cleaves proteins with enzymatic activity by recognizing proline and alanine with deposition of the polypeptide N-terminal chain. CD26 DPP4 is a homodimer, a glycoprotein, bound to the cell membrane. The expression of angiotensin II of ACE, angiotensin II receptor type 1, AGTR1, and bradykinin receptor B1, BDKBR1 increased with age, especially in cardiomyocytes. So the elderly are more attackable because they have more locks, multiple ports through which the virus can penetrate. In cardiomyocytes, ACE2 converts vasoconstrictor angiotensin II into vasodilator angiotensin 1,7 and converts the DESR9-BRADYKININ promoting peptide into inactive bradykinin 1,8. Internalization of ACE2 by SARS-CoV-2 potentially neutralizes the function of these metabolites in the renin-angiotensin-aldosterone system, RAAS, which regulates blood pressure and electrolyte balance. Bromelain, isolated from the stem of the pineapple, bromelain can inhibit SARS-CoV-2 infection by targeting the three hosts ACE2 and TMPRSS2, transmembrane serine protease 2 and SARS-CoV-2 S proteins, lowers the expression of S-ectodomain. Spike on nicotine receptors. The spike protein also binds to the alpha-7 and alpha-9 neurological nicotinic receptors, blocking the intestinal and pulmonary anti-inflammatory function. This means that the viral and vaccine spike protein interfere with the receptors of the nervous system. In addition, nicotinism receptors are also present in the brain so that the damage felt at the neurological and psychic level after the disease or after vaccination can be understood. Interferon. Viruses use cellular defense mechanisms to their advantage. An example is with the activation of the interferons produced by the body to enhance the immune defenses against viruses. Interferons genetically promote the synthesis of ACE2, hence the locks that allow the virus to enter. This explains the aggravation in patients who had been administered interferon with the hope of fighting the virus. In reality, it provided new locks to be able to enter. What's dangerous? What does it aggravate? This part is for understanding points 3 and 4. The syncytium, coagulation, mimicry, fever. What to avoid? Cellular syncytia. With syncytial viruses and CoV-2, the cells fuse together, giving rise to a conglomerate, syncytium, from which the name derives. When the cells come together, they create clusters that can obstruct the circulation of blood, oxygen flow and the functions of the various organs. These abnormal syncytial cells are generated by the ability of the coronavirus spike protein to stimulate fusion between infected cells and neighboring cells. Research shows that spikes activate a family of cell proteins, called MEM16, which are essential for cell fusion. This mechanism also underlies platelet activation, and could therefore also explain why 70% of patients with severe COVID-19 develop thrombosis. So both the virus and the vaccine that makes our cells produce spikes can activate cell syncytia and cause thrombosis to die. Niclosamide counteracts this effect as well as Plaquenil. The virus and coagulation. In 2020, Professor
Salvatore Spagnolo
has highlighted, thanks to his experience as a cardiac surgeon, that the virus produces a coagulation in the pulmonary vessels and capillaries. Coagulation prevents the flow of blood into the alveoli and therefore the possibility of carrying oxygen from the alveoli to the body. He therefore understood that it is essential to keep the blood fluid and prevent clotting. It was ignored and deliberately opposed by AIFA. This article was published in April 2020. From this article it emerges the primary importance to prevent the formation of coagulation and to counter it in all ways. Povidone iodine, like zinc, obstacles the entrance of the spike into the cells. These articles were published in 2020 in America, Elements of Prevention Against the Coronavirus and Hypothesis of Iodine-Dependent Immune Collapse in Coronavirus Microclots, Microvascular COVID-19 Lung Vessels Obstructive Thromboinflammatory Syndrome. and this hypothesis of protective effect anti-COVID-19 of iodine and or hydrogen peroxide garglings and mouthwashes to prevent infections through oral mucosa. Curiously, this latest article has disappeared from the internet tab, as is. The interesting thing is that a research subsequently appeared which states that with povidone iodine, healings are effectively obtained with an 88% reduction in deaths from COVID. Molecular mimetic, the viral spike and therefore also the spike produced with vaccine gene therapy. They have sequenced homologies with numerous cells. The chemical analogy, the similarity between the spike and our cells, it can trigger reactions against ourselves as well as with the virus because the immune system confuses us and the virus, for which, for molecular mimicry we will have interstitial pneumonia for the analogy with the alveolar walls. Vasculitis, thrombosis, can also depend on molecular mimicry. Heterological immunity. There may be reactions of heterologous immunity with the virus and with the RNA vaccine. The vaccine can reactivate heterologous immunity with persistent endogenous DNA viruses. The antigen is a spatial configuration. SARS is able to cause an antigenic representation of endogenous viruses already present in the body. For example, cytomegalovirus is related to vascular accidents, as the endothelium has similar antigens. In addition, the following can be called into question. Sixth disease, chickenpox, mononucleosis, danger. An external stimulation can produce a reduction of TREGs with activation of cytotoxic T and activation of autoimmunity. The vaccine activates type 1 interferon, which promotes local systemic and autoimmune inflammation. Fever and glutathione. To defend itself against viruses, the body tries to inactivate them by producing heat. Fever has this function. Up to 39 degrees, fever is a powerful ally. Glutathione is also used to fight viral damage. The drug that blocks these two powerful body defenses is paracetamol, known as teicoplanin. It is understood that teicoplanin could be a cause of the dramatic worsening seen in patients. In fact, from our observations, those who did not take it quickly resolved the viral infection. The glutathione, GSH, is the most powerful antioxidant in existence and is naturally produced by our body. It is composed of 3 amino acids, glycine, glutamate and cysteine, and has a hepatoprotective function, that is, it protects the liver, which is our purification unit, from oxidative damage, therefore also from the damage of an incorrect diet and from viruses. This antioxidant participates in processes ranging from the production and synthesis of proteins to tissue repair, up to the participation in the synthesis of molecules of the immune system. As a hepatoprotector, therefore as a protector of the liver, glutathione collaborates in the purifying activity. Glutathione nutrition. Useful foods: cabbage, cauliflower, broccoli, asparagus, garlic, onions, leeks, but also whole grains, meat, fish, dairy products, avocado, watermelon, spinach, peaches, apples, melon, grapefruit, carrots. By regularly consuming these foods, it is possible to stimulate the production of glutathione peroxidase, which is the antioxidant that causes its beneficial effects. It is advisable to combine these vegetables with foods rich in selenium, such as fish, anthocyanins, such as red-purple fruits and vegetables, from cherries to strawberries, from oranges to grapes up to red pepper, with proteins and high biological value, such as meat, fish and eggs. The genes, an innate immunity molecule, called mannose-binding lectin, MBL, binds to the spiked protein of the virus and blocks it. The point is that genetic alterations of this molecule, which modify its behavior, may be linked to a greater severity of COVID-19. In 15–20% of severe COVID-19 cases, a genetic disorder does not cause interferon to be produced or destroyed. The virus promotes an important release of type I and 3 IFNs and inflammatory cytokines and chemokines, i.e. molecules that act as mediators of natural immunity and inflammatory response, known to contribute to the cytokine storm observed in COVID-19. It was interesting to observe that type IIFN, released by plasmacytoid dendritic cells, PDC, is able to stimulate the antiviral response in infected lung epithelial cells. The immune system, innate aspecific system, it responds in a nonspecific and generalized way to invasions. This defense system solves 90% of the problems caused by contact with bacteria and viruses. Cells, mast cells, eosinophils, basophils, macrophages, neutrophils and dendritic cells, adaptive exquisite system, respond specifically and targeted to invasions. Possesses an immunological memory. Cells with immune memory, B and T lymphocytes, TREG or helper and cytotoxic. Once the battle is over, they remain in the bone marrow ready for new attacks. Humoral B lymphocytes release antibodies into the blood against viral proteins such as spikes. The humoral immunity that is maintained over time, with a high antibody titer, is defined as sterilizing immunity since it allows us to avoid a second infection, as the virus is blocked by neutralizing antibodies. T cells like sentinels scour the body for viruses. The iTreg. They are more efficient in fighting viruses. Many are cross-reactive, that is, they react to several types of viruses at the same time, such as influenza, and also to many mutations. Pre-existing contact with a flu virus may have immunized against SARS-CoV-2. Cytotoxic Ts scan the body looking for infected cells to eliminate them. The lymphocytes, friends enemies. Our defense system uses various cells. The first to intervene against viruses are APC cells, antigen-presenting cells, which present the viral antigen to lymphocytes complexed on their membrane with the proteins of the major histocompatibility complex, MHC, so that they develop specific weapons against them. The dendritic cells presenting the antigen are those that most activate the T, glymic lymphocytes. They capture protein antigens, peptides, with two possibilities. Those that have penetrated into the epithelia, transport them linked to the MHC through the lymphatic system to the lymph nodes. If, on the other hand, the antigens are encountered in the blood, they are transported to the spleen. B lymphocytes are able to recognize antigens directly, thus acting as APC on the membranes of the host cell, and produce antigen receptors also in soluble form. They have a short life. T lymphocytes have a system of receptors, DCR, CD4, CD8, through which they are able to recognize the antigenic peptide present in a complex with MHC proteins on dendritic cells, B lymphocytes and macrophages. They survive for years. Lymphocytes are smaller, more specialized cells. With COVID, we will have low levels of B lymphocytes, therefore antibodies, and T helper, TREG. In the elderly, there is a deficit of peripheral blood. Cytotoxic CD8 lymphocytes, monocytes and T killers increase, which explains the cytokine storm that attacks all organs of the person. Platelets increase with a greater presence in men of anti-interferon antibodies that could block the action of interferon itself. The lymphocytes, the lymphatic cells following aggression from the medulla migrate to the thymus and differentiate into Treg, helper, X suppressor, protective anti-inflammatory regulatory. Lymphocytes have CD4 receptors, which direct them towards dendritic cells, macrophages and B cells. They are long-term memory effectors. They have CD26 as a membrane marker. They are the allies against viruses. T cytotoxic T killer. They produce inflammation and autoimmune responses, have CD8 receptors with which they attack and kill nucleated cells equipped with foreign peptides such as those infected by the virus. They do not have the membrane CD26. These increase with age. CD26 is reduced in dying cells in apoptosis. CD26 is reduced in dying cells in apoptosis. To fight viruses, We need to increase anti-inflammatory Tregs. CD26 favors the production of Treg spike, and therefore the vaccine that produces the spike blocks CD26 by inhibiting the production of anti-inflammatory Treg cells. The anti-CD26 homotoxicology should block the CD26 receptor, preventing the coupling of the spike for competition, and produces protective Tregs. The homeopathic anti-CD26 simulates the pathogenesis of the lack of synthesis of Treg. Viral progression. The virus begins to replicate and induce the death of epithelial cells, also due to pyroptosis. Pyroptosis is a process of cell death associated with the production of interleukin-1 beta, IL-1 beta, and interleukin-18, IL-18, two cytokines produced by the immune system, highly inflammatory and capable of exacerbating the state of inflammation linked to entry of the virus. At this point, two different pictures are generated. Improvement. Inflammation leads to the recruitment of CD4 and CD8 cytotoxic T killer T cells, to the production of neutralizing antibodies and, therefore, to a protective response. In this case, the cytotoxic lymphocytes, neutralizing antibodies and macrophages work together to eliminate the virus, kill the infected cells, and the patient undergoes recovery. Worsening. In the elderly, a neutrophilic infiltrate prevails, and CD4 T lymphocytes began to produce cytokines which, instead of supporting the immune response, amplify inflammation. In this case, the CD4 and CD8 cells undergo a depletion process and begin to express molecules on their surface that block their functionality. In particular, dendritic cells are the starting point of the immune process. The viral antigen penetrates the dendritic cell, which fragments the antigens and presents them outside its membrane. The dendrites allow the T lymphocytes to recognize the antigen presented and to differentiate as CD4 and CD8 cells. CD8s differentiate into CD8 effector lymphocytes. CD4s proliferate by developing long-term cellular memory, which is what we need to enhance TH lymphocytes. T helper, D4 or CD4, stimulate and support the recognition and response action of T and B lymphocytes, favoring their differentiation into plasma cells and the production of antibodies. They are the elective target of the AIDS virus. Regulator T regulators have CD25 and FOXP3 markers. When the T lymphocyte expresses CD25, the cells are CL1'd against the immunogen. Positive swab equals external virus. The virus enters people from the respiratory tract. For this reason, swabs are made in the pharynx because it is the antechamber of the respiratory tract. But first of all, we need to understand the concept of out of the body and into the body. Outside the body is everything that is external to the protective membranes, therefore the skin and mucous membranes. Outside the body, it is both what surrounds us and what is inside the digestive or respiratory tract, because it has not yet crossed the barrier of the protective tissues. In the throat, in the nose, in the intestine equals out of the body. Inside the body is the internal part of the membranes that delimit us, therefore inside the skin and mucous membranes. In the cells, in the matrix, in the organs equals inside the body. A virus on the skin of our body is external, it has not yet entered. The same is true for a virus in the throat, it is still external, it has not yet penetrated into the cells of our organism. When a swab detects the presence of substances in the throat or behind the nose, it means that there is something in that area. It does not mean that the person is sick, it means that there is something outside the body. It is in our pharynx, but it is outside the body. It is still external, it does not mean that it has already entered the body. So a positive swab does not mean disease in progress. If the person has no symptoms, the virus is probably passing through, and with the surface immune defenses, the body eliminates it. Immunoglobulins A, IgA placed on the digestive and respiratory mucous membranes, are the soldiers that hinder the entry of the virus through the digestive and respiratory membranes. Unfortunately, IgA is not stimulated by vaccinations, so we have to support the body in a different way. It is understood that gargling 5 parts water with 1 part hydrogen peroxide helps the body mechanically fight viruses in the pharynx. The same applies to gargling with povidone iodine formulation, as iodine prevents the virus from entering the cells. With positive swab and no symptoms, it is useful to check after 2 days if you are still positive to differentiate if it was a randomly passing virus or a residential virus. It can also happen that the body has produced cellular waste called exosomes, which are similar to viruses and which are mistaken for virus testing, so caution and discernment is needed. Why do some people get seriously sick? They found that those who are seriously ill have a genetic defect in the production of interferon, the sensor molecules that activate the receptors of the immune cells called TLR, which have the task of warning of danger, are not produced. Unfortunately, the vaccine activates type 1 interferon in everyone, which favors local systemic and autoimmune inflammation. So all vaccinated people will have unpredictable modulations on this precious cell defense molecule. How to know when does the disease get worse? The tests show that interleukin IP10, 10 and interleukin 6 increase. Other indicators are C-reactive protein, ferritin and D-dimer, the decrease in Treg, basophilic granulocytes and plasmacytoid dendritic cells also decrease. Lack of vitamin D. It has been observed that a lack of vitamin D promotes aggravation. High doses of vitamin D prevent aggravation and promote healing. Metabolic syndrome. Lipids in circulation. The excess of carbohydrates creates an insulin disorder that favors the deposit of fat in the cells and especially in the abdomen. It turned out that people who have severe symptoms with death, it is due to the excess of fat cells that release fats that end up in the pulmonary alveoli forming a membrane, hyaline membrane, which prevents the passage of oxygen from the alveoli to the vessels. Naturally, the inflamed visceral tissue releases the inflammatory cytokines and free radicals that favor interstitial pneumonia, DIC and fatal cardiac arrhythmias. A low-carbohydrate diet or fasting, in addition to physical activity, are protective. Metformin, which promotes the metabolism of carbohydrates, may be useful, particularly to be considered after receiving 2 or 3 doses that inducing vaccine immune erosion or post-vaccination immune deficiency. Mutations. Having vaccinated during the pandemic against the opinions of scientists, numerous viral mutations have been activated. Some are less harmful, others more dangerous. Having found the presence of segments of HIV in the virus, it is possible that aggressive reactions induced by this component are activated. Ivermectin has shown a certain degree of protection. Resources. Epigenetic naturology. Prevent entry into mucous membranes and cells. Prevent clotting, which can trigger a domino effect. Counteract inflammation. Support Treg immunity. Protect cells and vessels from mimicry. Favor the immune hunters. Naturological epigenetic immune synthesis. What hinders the entry of the spike? Iodine povidone, zinc, bromelain, H2O2. What hinders cellular locks against the virus? Anti-CD26, anti-CD25 OTIA, Th2 homeopharm. What hinders the formation of cellular syncytia? Niclosamide, Yamsin, hydroxychloroquine. What hinders coagulation and protects the vessels and capillaries? Acetylsalicylic acid, heparin, bioflavonoids, blueberry tajenzindiflin, anti-CD25 and 26 OTI. What to compete with the nicotinic receptors, apolifunctional symbol alpha 7 and apolifunctional symbol alpha 9, D-vitamine, omega-3, melatonin. Spigeliacidicolin. What improves expectoration and stimulates the defenses? Glutathione NaCN. Acetylcysteine fluimucil. What C? Counteracts inflammation? Acetylsalicylic acid, bromelain, ananase, ivermectin. What to recover the taste? Nicotinic receptors. VIT-D 30,000 units per day. Homeopathic spidelia. What can prevent molecular mimicry? Anti-CD13 OTI, viral RNA and DNA with immunity. Heterologous, cytomegalovirus, elderly. Homeopathic sulfur, atrial fibrillation. Dexamethasone nitrocamucidum, chlamydia. Calcarb, fevers, metabolism. With counteracts the metabolic syndrome, free radicals in the cytokine storm, reduction of carbohydrates and increase of antioxidants against a sore throat. Rosemary or mountain pine essential oil against intestinal disorders. Rosemary essential oil, charcoal. Thank you for your attention.